Thank you. It's a great pleasure to have an opportunity to speak to this group. I sincerely wish I were there because the topics that I've been hearing discussed are entirely within my sphere of interest, and it would have been great to have an opportunity to discuss these issues with you. I'm going to um, move. Let's see if I can. This seems to be frozen. Ah, okay. Let me go back. Um, First of all, I'd like to emphasize that um, we are in have been in two pandemics, and pandemics are part of human history. There will be more pandemics, and it's important that we have all the tools that we have available to be used to protect human health. Um, I will speak about cholera. I've been studying it since the 1960s. It's a water-related disease. We're in the seventh pandemic. It occurs in about 50 countries. We have ascribed the Bengal Delta as the homeland, but it's quite unfair because 150 years ago, cholera was rife in the US, Europe, Canada, South America, globally. Cholera bacteria exist in the aquatic habitat, and I'll make the case for that today. Um, and the evidence of the new biotypes is clear that we really can't eradicate the disease because the bacteria carry out functions in the environment. The bacterium is easily cultured, shown in the petri dish to the left. With appropriate dyes, the selected colonies can be obtained. The bacterium is a polarly flagellated um, bacterium, and the disease is essentially an extensive loss of body fluid. The work that I will describe really began in 1960 as part of a PhD work at the University of Washington, and then subsequently work in the Chesapeake Bay, where we clearly were able to establish over about 20 years the um, fact that the bacteria are associated with plankton, zooplankton, copepods to be specific. What has not been emphasized and should be is that all Vibrios, at least all that have been tested to date, all species, are chitin digesting. And in addition, we have been able to demonstrate that Vibrio cholerae cells actually contain genes coding for bioluminescence, sort of evidence that clearly these are environmental microorganisms native to the aquatic environment. They can be isolated from other marine animals and plants, but in fact, their vector I like to ascribe is the copepod, which represents about 95% of the zooplankton population of the world oceans. We developed the first model from the Chesapeake Bay work, namely that in the spring, with warmer temperatures, increased sunshine, the burst of the phytoplankton is followed by the zooplankton, which feed on the phytoplankton. And then with the collapse of the plankton blooms, there is released into the water column from the zooplankton the bacteria, which then, if the water is not treated and ingested directly by humans, is a means of transmission of the disease. So we established back in the 60s and the 70s temperature, sunlight, rainfall, pH, dissolved oxygen, salinity, and nutrients as important physical chemical characteristics associated with the cholera outbreaks. We were criticized severely back in the late 60s, early 70s, that the model had been developed for the Chesapeake Bay, but it would not hold in Bangladesh or in other countries of Asia. So that was a challenge. We went to Bangladesh in 1975 and began our work. And indeed, person-to-person transmission clearly is a mechanism of transfer, but the environment really is the source, and the zooplankton and the presence in the environment was documented. What really was interesting was in 1985 when Landsat was launched, the satellite was able to measure chlorophyll, the various types of chlorophyll, and sea surface temperature and sea surface height. Because of this relationship with plankton, it occurred to us that indeed we could use satellite sensing as a predictor of risk of cholera. And so we did our first experiments between 1985 and 1995 and then published a paper in 2000, 24 years ago, showing the relationship of the outbreaks of cholera in the Bay of Bengal, where we measured by satellite a mile quadrant right off the Bengal coast of the Sundarbans of the coastal area of Bangladesh. And then because we were doing work with the medical laboratories and the hospitals in the Sundarbans, we we counted the numbers of cases. And so you see the red line are the cholera cases in the Sundarbans in Bangladesh, and the blue line is the sea surface temperature, and the other parameters of chlorophyll, etc., were correlated as well. So that was the first relationship of the potential of at least for us, for the use of Earth observation as a means of a public health tool. Um, we went back to data that had been available in the British um, Museum and Science Facility in London to obtain the data from India from 1875 to 1900, and in particular, the villages around the area of Delhi in India. And we were able to show the relationship of the odds of cholera for temperature and rainfall. And we were able then to extrapolate from what occurred in Haiti in 2010 when in the spring the earthquake, very severe, very high intensity earthquake with a great deal of damage. And then in the fall, in September, October, November, cholera broke out extensively, one of the worst cases at that time. And we were able to show by downloading data from satellite observation that the air temperature in 2010 was the highest in 50 years. And similarly, the rainfall that occurred in August, well, around September, October, when a hurricane went through Haiti, was the most severe, the heaviest rainfall in 50 years. So again, these correlations of environmental parameters were critical. We've been able to sort out cholera epidemics into those that are epidemic, such as occurred in around Delhi uh, every large, let's say 50-year interim, when there was a severe rise in temperature, a gathering of people for festivals of some sort, and poor sanitation. That mix ends up with the epidemic cholera. The mixed mode cholera is that which one observes in Dhaka, where there's an influx of the coastal waters with plankton, and at the same time, um, peaks that are associated with um, gathering of a population at given times. And then along the coast of Bangladesh, endemic cholera because it persists throughout the year, and there are spring peaks and fall peaks, but throughout the year. So we were able to develop this hierarchical model for the transmission where favorable abiotic conditions, blooming of phytoplankton that promotes the Vibrio survival and food for zooplankton, and then the proliferation and association with zooplankton and crustaceans. And these are the various parameters uh, associated with the climate variability that are drivers of epidemics of cholera. So the timeline is that establishing temperature, rainfall, salinity, nutrients, and other abiotic factors in the 60s. And then we were able to determine in the 70s the phenomenon where, since the bacteria, the Vibrio bacteria, are associated with zooplankton, the zooplankton in many parts of the world, particularly the bays like the Chesapeake Bay, go in the winter months, burrow into the sediment and go into a state of dormancy. And the bacteria, being commensal with the zooplankton, go into this non-culturable state. And that explained why the WHO investigators were unable to isolate it during the winter months from any of the vectors, perhaps cattle, etc., because the bacteria present in the environment were essentially in a dormant state. And then additional characterization in the 90s, and then uh, exploration with Earth observations starting in the 2000s for cholera. And we're able to do essentially a real-time prediction, which I'll describe in a little bit more detail, using predictive intelligence systems, computational models, along with observation for waterborne pathogens to provide a risk prediction in order to beat the curve, so to speak. That is, to wait to when cases begin to appear, is important to have medical supplies, safe water, rather than at the time when the epidemic is clearly obvious. And so being able to predict early to allow this intervention is really what the goal of the research is. And so we've been able to improve the um, prediction model for cholera um, with the new information that we've been able to gather, and the ability then to do analyses globally, um, and more importantly, to move beyond use of Landsat to some of the more sophisticated and recently declassified observational satellites that provide us with um, more accurate and more extensive sets of data. We tested the um, model that we had about 6 or 7 years ago when the outbreak occurred in Yemen. We did this was a very severe outbreak, probably the worst to that point of cholera anywhere in any country in the world. And so we did a retrospective analysis of what was occurring in Haiti and then did our prediction. And based on our predictive model, it was this area that we determined would be the severe um, epidemic form of cholera. And indeed, the actual results, the epidemiological map showed that is where it occurred. So we published this as a quick note. It was picked up by Scientific American, and then a British scientist, an aid worker with the British Aid Agency, Fergus McBean, got in touch with us and asked us in the June of the spring rather of 2018 to provide risk maps for the following year, that is for 2018. And we were able to do that, at least at that time, 8 weeks out, and we were able to provide risk maps. We are now able to provide and continue to provide 4-week risk maps every 2 weeks. And what we were able to observe is that the risk scores in that early work in Yemen showed that, in fact, we were really hitting the mark reasonably well with our predictive model. And so in 2018, we were able to show where the risk was highest, and we were able to moderate or at least contribute to the reduction in the cases of cholera, because it was then possible to provide physicians, healthcare, safe water, specifically geographically to where the need was the greatest. And so the performance of the cholera prediction in 2017 to 2019 showed it was about 60 to 70% in sensitivity, specificity, precision, and accuracy. We've now been able to raise it to over 90% with the increased data and analysis. And this is one of the latest analyses which we provide. This was a year ago, a couple of years ago. Um, and we continue to provide um, every 2 weeks risk maps for Yemen, and we now include Ethiopia and several other countries in Africa. So we were able to provide risk maps for Ethiopia, Afghanistan, and even some work in Ukraine, where we're able to show some areas where the risk would be highest, particularly where the war had resulted with Russian destruction of the water purification plants and sewage treatment plants. And with the climate and the population, coastal populations, we were able to point out the risk, particularly in Mariupol, and provide that information to the UN. Um, we're continuing these risk analyses, and what was particularly interesting in Malawi in June, we were beginning to find signals for cholera and alerted the UN agencies. And by late summer, there were cases appearing. So the predictive intelligence um, seems to be a powerful tool for public health. We've done some analyses of the Gaza region, and the risk, interestingly, is highest in the neighboring country of Egypt, where many of the um, um, migrants have moved. Um, and so this then is the um, risk prediction as of November, December for cholera in that region, and we're continuing to provide analyses. Um, what I think is important to point out is that these are risk scores and are dependent on the availability of safe water and certainly health systems. Um, what I think is important to understand is that, again, we want to be able to intervene to prevent cases, and so perfection of the work that we're doing continues. Um, we provide a prediction hub. It's online, and this is accessible for cholera, but also we're moving now to understanding other vibrios and other diseases. It's important to point out that 50% of the world population lives within 50 miles of coasts, which means what happens along the coast is critical to human health. A study that we did with British Marine Laboratories, Plymouth and Southampton, several years ago, published in 2016, 8 years ago, in collaboration with a group, Dr. Carla Pruzzo and her team in Genoa, Italy, with the Max Planck team and the British Marine Laboratories and ourselves in Maryland as a team. Um, we took advantage of the samples collected of water and plankton by the British Marine Laboratory some 50 years ago. Over the years, we then were able to go to the plankton samples, extract the nucleic acid, and then probe for Vibrio parahemolyticus, the pink Vibrios, Vibrio, I'm sorry, green, Vibrio vulnificus, pink, and then Vibrio cholerae. And we were able to show that with the warming of the sea surface temperature, there is an increased numbers of vibrios in the plankton associated with the plankton populations that are triggered by temperature. And we were able to relate this then to the outbreaks of Vibrio parahemolyticus, Vibrio vulnificus, and occasionally uh, Vibrio cholerae. So that this was perhaps the first correlation of a climate change parameter, sea surface temperature, associated with actually human um, cases of Vibrio-driven disease. We've now been analyzing the Vibrio perihemolyticus and vulnificus because they are the the causative agents of infection in the U.S. Uh, about 95% of waterborne seafood-related deaths are caused by Vibrio vulnificus in the U.S. and about 80,000 Vibrio perihemolyticus cases, 65% of them are foodborne. So these are important in the U.S. And indeed, we've been able to determine, these are two PhD theses, Dr. Carl Brumfield, who graduated just this last summer, and another student, former student, Dr. Montardo, who she finished her PhD just a month or two ago. And they have been working um, on the Vibrio infections. Michelle has shown that gathering the data that clearly there's been an increase in Vibrio infections since 1996. The work that Kyle has done has shown that where normally there has been a peak of Vibrio infections around August, September, it now has broadened with the climate warming, so that beginning in June, we start seeing, May and June, we start seeing Vibrio infections all the way through December. This long-term increase of vibriosis in Maryland includes Vibrio cholerae, Vibrio fluvialis, alginolyticus, vulnificus, and perihemolyticus. In fact, the Centers for Disease Control, CDC, in the US has now decreed any Vibrio is a reportable pathogen. So the Vibrio has become then now part of the monitoring of microorganisms. The work on seasonality that I had mentioned, um, the work of Carl Brumfield's PhD thesis in the lab, has shown the seasonal broadening due to warmer temperatures of the Vibrio infections. We have also been continuing satellite monitoring of the um, temperatures along the coast, following it and calculating risk probabilities, risks, scores for the coastal regions of the U.S. And we began to see that along the eastern seacoast, up around New York, New Jersey, Connecticut, the risk was increasing. And sure enough, sadly, this last year, there were 3 deaths, 2 in New York and 1 in Connecticut, from Vibrio vulnificus associated with exposure to seawater and undercooked seafood. Then, with the extreme warm temperatures in Florida, we've been doing a good deal of work because we collaborate with a former postdoc, Dr. Antar Jutla, who's a tenured associate professor of engineering in the civil engineering in the College of Engineering at the University of Florida. And so we've been doing sampling and analyses there and have now a trend map on the left and for vibriosis in general, and then for Vibrio vulnificus cases in the right map. And you can see that some regions along the coast have very high risk of Vibrio vulnificus potential. In fact, the long-term increase of Vibrio vulnificus cases in Florida have been significant, dramatic in fact. Um, Interestingly, we've been able to show cases, and fortunately, deaths have not been have been rising, but because of the awareness of the medical community now, there is a, a rapid treatment of potential vibriosis for patients coming into the emergency room in Florida. Um, see if I could move this forward. I think I'm sort of, oh, there we go. Um, we then followed the Hurricane Ian, its course through Florida, and where the most severe winds and greatest damage occurred. We took some samples from that area. Um, these are um, hatchery and fishery plants, but we took our samples in the water areas nearby, and these samples of oysters and water were collected in October of 2023, and we were using the technique for shotgun sequencing that allows us to um, match up sequences from extracted DNA to match up with our database that allows us to identify all the microorganisms, bacteria, viruses, fungus, protists, as well as the genes for antibiotic resistance and pathogenicity for microbial characterization. We've been doing this in studies of individuals in a collaboration with Calcutta, India, where they did the classical analyses and we got the DNA from the stool samples and we're able to characterize very precisely the disease agents that were involved, where they could isolate Vibrio cholerae. We were able also, from the DNA from blinded samples, to determine those with the Vibrio cholerae. But what we found was that when they were not able to isolate Vibrio cholerae, but the patients had symptoms of cholera, that there were other bacteria. And we were subsequently able to show that in a comparison that the healthy population carries a large number of Vibrios compared to the Western human NIH downloaded database. Now, I just mention this because we use this technique to analyze the samples that we've collected off the coast of Florida and in the Chesapeake Bay. And we are finding that there are many Vibrios in those samples that we've been collecting. And um, the Vibrio species include not just Vulnificus, Parahemolyticus, but a variety of other Vibrios, even Vibrio cholerae. And so we're now in the process of analyzing the genetic composition, and we've been able to identify the presence of antibiotic resistance genes and other pathogenic coding genes in the populations. And so this provides us then with lessons that we can learn from what we have done in studying the Vibrios in the various parts of the world, Bangladesh, east coast of the US, in the North Atlantic, etc. And so with these environmental parameters, we've been able then to develop then theories of the vector and transmission of the pathogen and transmission to the human population. We've carried this information to COVID. I don't have to describe the symptoms. We all are far too familiar with COVID. But what is interesting is that there are environmental parameters, ambient air temperature, humidity, um, the movement of populations that can be monitored again by some of the satellites, the sensitive satellites, and also using cell phone data that are publicly available. And this allows us then to fit into the model we've developed for cholera, the data appropriate for COVID, and then to provide a predictive analysis of risk of COVID. Because we're able, from the DNA work that we've been doing as well, we have been able clearly to identify the SARS-CoV-2 virus and its variants. And that allowed us then to do what many others, in fact, I think one of the earliest publications came out of Italy on the use of wastewater for detection of the COVID virus. Not a new phenomenon because we had been using it for polio, detection of the polio virus some 40 or 50 years ago. But this has turned out to be a very important community analysis of community health. And we were able then, in our own work in Maryland, starting in March of 2020, when the outbreak of COVID was just beginning in the US, we started analyses from June through the end of July for this example was one city, Frederick, Maryland, and we're able to show with the blue, the CDC recommended genes to be tested for, and the orange are the additional genes that we did to be absolutely sure we were picking up COVID. And we were able to show a peak in the wastewater, and then the cases began to occur, as has been shown by many other people in other countries, other states of the US, this correlation of the COVID virus in the wastewater and the outbreak of community cases. And like others, we were able to show that by sampling the dormitories in St. Mary's University in Maryland, we were able to detect a single dormitory where they had 221 students resident, 10 were positive, 9 asymptomatic, and they were able to quarantine the building, the students in the building, and then prevent having to close down the campus. This is familiar to you all from the work going on. We have continued to profile using the DNA metagenomics because this has been, I think, a powerful way for community public health that can be tied with the satellite observation. And this is a form of ground truthing, perhaps to make it more quantitative. And so we applied the satellite sensing and the modeling with the data for COVID and produced our first risk map in April and May of 2020. And we have continued in 2021 and to the present to be able to provide risk maps. The upper is our risk map, the lower are the actual cases. This was for 2021. We now provide uh, uh, a monthly risk map that um, county by county for the risk of COVID in the US. We're in the process at the present time of trying to bring together the wastewater data with the risk map from the satellite observation. Very crude, very preliminary, but I think it's probably a way that we can pursue the potential of quantifying the Earth observation with cases based on the ground truth measurement. Here, the cases are in yellow. The um, daily new cases are a dotted line. Um, the N of the copies per mil, this is one aberrancy. And the red are the risk map, risk calculation by satellite. And so it's very crude, very early, but we're also able to tie this with antibiotic resistance and virulence genes so that we can not only determine the presence of the organism, but we should be able to correlate with the potential pathogenicity and other characteristics. So this is essentially where we are at the present time and where we hope to be going. So we have a Vibrio prediction hub, a COVID prediction hub, and clearly this is very possible for other, especially waterborne diseases. And I'll conclude by this presentation of 60 years of research, starting back in the 1960s to the present. The words of John Muir, the founder of the Sierra Club, an environmentalist. When one tugs at a single thing in nature, he, and then I've inserted and she, finds it hitched to the rest of the universe. And so many collaborators and colleagues from the International Center for Diarrheal Diseases Research in Dhaka, especially Dr. the director, Dr. Munir Alam, and others. And then in NICED, the Kolkata laboratory, Dr. Balakrishna Nair and Dr. Ramamurthy, former directors of that lab. Many students from the University of Maryland, especially currently Kyle and other students, and colleagues from other universities, other countries from around the world. It's been really a UN operation, and I'm very proud of the collaborations we've developed. Much of this is written up in a book I wrote a couple of years ago, A Lab of One's Own. And yes, Vibrios are everywhere, even in Iceland. We isolated them from the thermally heated waters of Iceland. They've never had cholera, but the Vibrio cholerae are present. So with that, I'll close and stop sharing and be very happy to take any questions that you would like to present. It's been an honor to speak to you, and I really wish I'd been there because everything I've been hearing discussed is so interesting and of so personal interest myself. So thanks again for the opportunity. If
Thank you very
you have,
much,
sure,
Professor Colwell, for that wonderful walk through some 50 years of Vibrio research and other emerging angles to your study and activities. It's an amazing piece of work built over years of hard work and innovative thinking. Thank you very much. We now have time for questions. There's one online. We can begin with that.
Sure.
Could we share that? Can Professor Colwell see it? While they are finding the question okay, here it is. While that one is coming up, may I ask a question to set the ball rolling?
Sure.
Over the years, when I've read the literature in this field, what's occurred to me is that diverse drivers and causative factors were identified for different regions. But it seems to me that your work now is beginning to unify the different approaches that were used for different localities and different situations. Would I be right in saying that you have actually reached the holy grail of generalization?
Well, I wouldn't say holy grail, but I think we're moving very strongly in that direction, and that's from what I'm hearing from the discussions that I was able to participate. I must confess, I did get up at 4 a.m. Getting up at 2 or 3 was a little bit much, but the discussion was excellent. The questions that were raised for improved data, we find that one of the biggest problems is getting the epidemiological data, the case data, from the local communities, particularly in the countries in Africa, where that would improve our risk prediction excellently. And so improved data and the correlation of the environmental data, I think what we have contributed and what others in your meeting, the meeting that you have in Rome, what you're contributing is the fact that finally we're able to get the medical community to understand the very significant role that weather, climate plays in human health. And it's very direct. And I think that holistic approach, not only of the individual taken ill, but the immediate environment and the general weather parameters play a major role in the spreading of the disease within the community. And so I think that it's the understanding of the holistic approach that the meeting in Rome and my work together is focused on. And I think it's that which we're contributing to another view of health and medicine. I hope that answers the question.
Yes, thank you very much. It is, I think, emerging as a game changer at any rate. Here is the question then, Professor Colwell from O'Brien, if I'm pronouncing it right, from ECDC to you. Apart from the 3 recent deaths in the USA, do you see a clear and significant spatio-temporal correlation with vibriosis case numbers and risk predictions based on sea surface temperature?
Yes, indeed. In fact, what I wasn't able to show is we've been doing work for the last 5 years in the Chesapeake Bay. And we are now preparing to release a Vibrio index for the Chesapeake Bay. And we can determine Vibrio vulnificus infection risk maps for particular parts of the bay where the salinity, the temperature, the nutrient, etc., these factors show a very high risk. And so this is essentially sort of like pollen count, if you will, for the asthmatics. This would be for the watermen and those who use the bay for recreation or who are harvesting from the bay for oysters and shrimp and crab, etc. Because we know that certain parts of the bay during the year are higher risk than other parts. So we're able then to provide correlation and a spatial temporal correlation with vibriosis, specific Vibrio infections in the Chesapeake Bay. And I think we are also considering providing risk maps for fishermen who are harvesting, let's say, oysters, lobsters, parts of, let's say, the Bay of Fundy, where the risk might be higher for Vibrio vulnificus as opposed to not having a risk. So I think we can use this for public health, but also for commercial fishing harvests.
Thank you very much. Do we have any more questions from the floor or online? Yeah. Willie. Oh, sorry.
Sorry. Thank you very much. That was fascinating. If I remember well, ESA supported a project in India somewhere looking at the use of automated wastewater sensors in, I think, the construction of a new digital city, and I think it was in Pune, if you pronounce that right. I don't know. Anyway, they were made in MIT, I think, and I'm wondering if you support, I mean, given your use of wastewater monitoring, I'm wondering whether you might see such automation as a way of reliably increasing the data flow to your sorts of studies.
I would indeed see the possibility. I wish I could take credit for the work you're commenting on, but I don't think it was us who did it. We who did it. But I have developed and over the years some of these rapid tests, and I think with the work that's being done at the various oceanographic institutions around the world, not just in the U.S., with DNA analyses. In water online, I think we're reaching a point where we can, in fact, have these very early warning systems that can be very precise and to the point. And what I think is most important is having the medical community understand the risk associated with, for example, Vibrio vulnificus. Not that they don't understand that having the infection is bad, but not being able to quickly recognize when it comes into the emergency room to treat it well and effectively, since its fatality rate is close to 50%. So I think it's being able to do the rapid diagnosis that is really important, and to have the risk analysis available for given regions to the individuals who live in those communities. I hope I've answered your question, because it's a very important one.
Sure, thank you very much.